when the cataloguing project wrapped up. Now, I should actually welcome the Morley family, who've got a number of people here tonight as well, and our southern members, shell clubbers. It's really lovely to see you, actually. So Margaret formally donated. I'll just shut that door so there's no echo from Hugh's computer. Margaret formally donated her collection to us in July 2016, just a couple of months before her death. First of all, I'd like to tell you a little bit about Margaret, the collector. Anyone who knew Margaret will already know a lot about what I'm going to tell you now, but it deserves retelling, as it just demonstrates why her collection was so valuable and why we were so keen to secure it for the museum. Then I would like to give you a project outline. I'd also like to touch on where we got our funding from. And then for the final part, I'd like to highlight what was achieved and the value the collection has brought to the Auckland Museum and its various stakeholders. So first, a little bit about Margaret herself. In the late 1970s, Margaret joined the conchology section of the Auckland Museum Institute, which is more informally known, of course, as the Auckland Shell Club or the Shell Club. She was also a research assistant at the Marine Department of the Auckland Museum from 1992 until her death in September 2016. She contributed a week for more than 22 years, and for us that is the equivalent of contributing over four full-time years of work. So that's a huge amount of work that she's contributed to a department that really actually only has 2 full-time staff members. She was a life member of the Auckland Museum Institute, and in March 2014, she received a museum medal for services to the Auckland Museum. And here at the bottom left, the photo is of Margaret receiving the medal with our former director, Roy Clare, and the board chair, William Randall. The conchology section of the Auckland Institute and Museum, as it was more formally known, of course, became one of a number of such divisions at the museum, and today it's the oldest and the longest surviving such section. And in its heyday, the club boasted more than 100 members. It ran monthly meetings, and it organised field trips and carried out coastal and harbour surveys, such as the Manukau Harbour Survey in the 1950s. It had its own journal, Puriaria, and between them, club members donated a huge number of both specimens as well as many thousands of volunteer hours. Margaret told me that she joined the club when one day in the late 1970s, she and Linda, who's here tonight, were at Auckland Easter Show, and they walked past a stand by the conchology section, and the stand was manned by the gardeners, Norman Knoll. And because the Morleys were a sailing family with a batch on Waiheke and some land on Great Barrier, Margaret thought that it would be an interesting hobby for her to take up because it was something that overlapped with their other activities. So Margaret took part in field trips organised by the Shell Club. Later, when she joined the Auckland Museum's Marine Department in 1992, she accompanied the department's staff during their organised field work, where she always helped with collecting and always helped with identification of marine specimens. And there were many more trips to the beach, either by herself or with the likes of Bruce. Margaret always came into the museum on a Monday, but never when there was a very low tide. Then she would have to be at the beach somewhere. Because the family, the Morley family owned a sailing boat, this was frequently the base for Margaret's collecting as well. And it took her to quite a few coastal places which were otherwise not very easily accessible. She also went on trips to far-flung places such as the Kermadec Islands, Chatham Islands, Stewart Island, the Auckland Islands in the Southern Oceans, and South Georgia and South America. As she decided to concentrate only on New Zealand shells, she also decided to specialize in micromolluscs very early on. Micromolluscs being those molluscs which are, as adults, reach no more than 0.5 centimeters, so 5 millimeters. And because of their size, they're really difficult to work on, as you need a microscope a lot of the time. And so often this is a somewhat overlooked and underrepresented group. Margaret meticulously recorded data and ecological information for the bulk of the specimens, and she worked with quite a few specialists such as Hamish, who's here tonight, and Richard Wilma over at Northern Territories, to have her data and her observations verified. And here are a couple of pages out of the catalogues which we got with her collections, and it shows the detail of her drawings and the sketches, the field notes and everything else. So it's a really invaluable resource. Many of the specimen lots and the data form the basis of publications, and these publications were put both in the Shell Club Journal Pureria and the Auckland Museum records, as well as her 2004 photographic guide to the seashells of New Zealand, which is the little book on the left-hand side. She was also a prolific artist who created hundreds and hundreds of black and white illustrations for scientific papers and bulletins and field guides and other publications. And she was quite happy to share these as well. And you can see how beautiful they are, you know, just from some of the ones that I've put on the slide here. So to summarize, much of Margaret's collections is the product of directed field work, and the data that it contains has great scientific integrity, which is why, of course, it's such an asset for the marine collections, as the marine collections are essentially research collections. So a little bit about the cataloguing project. When we uplifted the collection in July 2016, we estimated that it contained around about 8,000 specimen lots of molluscs, and that these were all exclusively from New Zealand. A significant proportion consisted of Margaret's micromolluscs, but the collection also contained another 4 discrete subsets, one from Waiheke, one from Stewart Island, one from the Chatham Islands, and these were all molluscs across all size ranges. And then there was also Margaret's general reference collection for the larger shells. Of course, as the museum's permanent staff is already fully committed to other work, whenever we accept collections, and particularly large ones such as Margaret's, we always have to bring in extra staff. And on the basis of a small pilot project we ran in 2017, we estimated that it would take a part-time technician 3 years to catalogue and integrate this many specimens into the general marine collections, and that this would require funding of around $90,000. So it was at this point that we approached the Auckland Museum Institute for help with some of this funding. The intention was to approach Lottery's Environment and Heritage Committee for the bulk of the funding, but Lottery's always requires that applicants come up with a third of the funding themselves first. So we made an application to the Institute for some help with this third portion, and they agreed to provide $12,000 from the Cheeseman Fund. And a further $18,000 came from the museum's temporary staff budgets. So together, this $30,000 and leveraged the remaining $60,000 from Lotteries. So we're very grateful that the Institute agreed to support the project, because on that basis, we were successful in our application to Lotteries. The project finally got underway in October 2018, and then despite some real impacts on it of COVID lockdowns, it was, well, near as damn it finished in June 2021, except for around 200 lots, which are being finished in-house. We employed Mrs. Jenny Riley, and Jenny was invited to come along tonight too, and I'm not sure if she's here, but if she is, hi Jenny. And we were really, really lucky to get Jenny actually, because Jenny worked at the museum as in the marine department during the 1980s and 1990s when she was a technician with us for 10 years. So she organised and went on many other field trips and knew Margaret personally. And so Jenny was exceptionally well qualified for this particular project. Despite several COVID-19 interruptions during the project, as I said, Jenny was able to catalogue, rehouse, integrate into our collections all of the micromolluscs, all of the Chatham Islands and Stewart Island shells, Margaret's reference collections, and all bar a couple of hundred shells from Margaret's Waiheke Island from her Aotearoa collection. So in total, Jenny catalogued nearly 7,200 specimens, which is not quite the estimated 8,000, but with approximately the 200 lots left, we're close to 7,500 lots. And if you add to that, that prior to the gift of her personal collection, Margaret catalogued over 3,000 specimens on the days that she came to the museum as a research associate. In addition, she identified nearly 8,500 specimen lots and went on nearly 900 field trips around New Zealand, either with museum staff, the Shell Club, or by herself. And from these trips, she deposited a further 5,816 specimen lots in Auckland Museum's collections, which before she even gave us her own personal collection. So in total, we now have well over 13,000 specimen lots in the museum, which we can attribute to Margaret. So why is Margaret's collection so important? Well, firstly, there are the micromolluscs, which are Margaret's specialty. These in particular added over 40 species, which were not previously represented in our collections, as well as adding some genera and a couple of families, which were not represented. And her collection boosted areas of the museum's collections where there were previously only very small holdings, so where we maybe only had one or two specimen lots at best. The collection also records the introduction of marine invaders to New Zealand, one such being the Asian date mussel, which was introduced in the harbour in the 1970s, and Margaret and Richard Wilma spent a considerable amount of time tracking this particular species and documenting it. Margaret added many specimens from difficult to access places like remote coasts, offshore islands such as the Kermadics, the Chathams, Stewart Island, Auckland Island. She also had records from deeper water material from trawls and dredging such as scampi research and marine reserve proposals, and sand reclamation projects such as the Pakari sand, which was added to a number of inner harbour bays such as St Heliers, which some of you will be familiar with. Likewise, she had access to material from other researchers such as Bruce Hayward's foraminiferal work and my own work on kelp holdfasts. Some of Margaret's specimens were part of informal longitudinal studies where she resampled areas over time, such as Aotearoa and the Bay of Islands and Paringaringa Harbour. So these will be valuable in future studies when we look at environmental change. Margaret's catalogues, of course, have a wealth of extra information on specific localities, on environment, life cycles, and at times they have, of course, those beautiful detailed hand-drawn illustrations of both the specimens and the maps of the areas where she collected. There are also numerous photos interspersed throughout her catalogue folders and published information and correspondence with other researchers. And finally, Margaret's collection information enhances the information we have in the museum for other collections. For other collections as well as collectors and collecting events. So it links with the ecology section of the Auckland Institute and with museum collecting and monitoring programs. and with historical collectors such as Powell and Marjorie Mistay and Captain Bollins, and with vessels such as the Lady Lisgard and fisheries research vessel Ikateri. She also reworked some of the old bulk sediment samples from the museum's collection, which date back to the 1920s and 1930s, from Powell's time, but even prior to Powell's time. And her collection incorporated material from other collectors who contributed to the museum, people such as Joan Coles, Norm Potter, Norm Douglas, Mary Whitehead, David Baker, and Dave Gibbs, and many other Shoal Club members. So in conclusion, this collection actually sits extremely well within the Auckland Museum's Marine Department, and that it not only adds to our own collections, but it fills in gaps, and it adds considerable depth to the historical knowledge we now have for the museum's collections and its collectors. And so I'd like to thank these organisations for supporting the projects. I'd like to thank you for listening and for bearing with me at the beginning. So when we finally overcame those glitches. So Bruce, do we take questions now if there are questions?
We're away. Okay, so I'm going to build on what Wilma's told you about and talk about Margaret Morley's research. So I'll use what she's been talking about as the introduction to what I'm going to be saying. So Margaret had on her wall at home or on the door of her study sign, "Forget housework, gone to the beach." Indeed, Margaret, although she did housework, wasn't very keen, and even so more, she hated going shopping. So any excuse she could find, if it was a fine day, she would head for the beach and avoid going shopping or doing the housework or other mundane things, she thought. So as Wilma said, this is Margaret's list of publications. She had one book, 40 co-authored peer-reviewed articles, and 70 articles in Poriaria and Geocene, as you can see there. Hope you can all read that. Don't worry. As Wilma said, she published one book on the seashells of New Zealand, first published in 2004, and had 3 subsequent editions because it sold so well. As you've noticed from Wilma's talk, Margaret was a fabulous illustrator. Here's some more examples of her work. On the right there, here she's got a reconstruction of a 20-million-year-old sabellid worm that's fossilized in the Waitakeris, and here she's reconstructed an ammonite as it may have looked. And over here she's holding a framed plate of foraminifera that she illustrated. Each one of these you might not realize is only 0.2 to 0.5 of a millimeter in size. And she's illustrated them all with pencil, and they were published. So this is Margaret's field gear. She always had a sun hat, a long sleeve shirt, 2 pairs of glasses, a small kitchen knife, hand lens, and a camera. And she also sometimes had a tea strainer and a large plastic bag for washing the bottoms of dirty stones, as she always liked to say. So Margaret was always on the lookout for marine invaders arriving in New Zealand. Probably a leftover from her time during World War II, when she was home in England, looking for invading Germans. But here in New Zealand, she was looking for marine invaders of the invertebrate or vertebrate type. And when they were she found some, she documented their spread and chased them all around northern New Zealand. Some of the marine invaders she worked on, as Wilma noted, was the Asian date mussel, but also the club tunicate, the small Japanese semilid bivalve, Theora lubrica, which I'll tell you a bit more about soon, Pacific oyster, the parchment worm, Echopteris, blue mussel, and the fierce Japanese swimming crab, Charybdis tonica. These are just some examples of the invaders that she noted coming into Auckland, and then recorded their spread around as they proliferated. One of the first works, as Wilma said, was the work on the Asian date mussel with Richard Willen. And she got very interested in this because they colonized her local beach, Buckland's Beach, and they formed big mats there at low tide. And you can see her here in Buckland's Beach in looking at them. She published on that in 1998, a report on the continuing study of 1988, study of musculister. And here, in a nicely hand-drawn map, accompanied that publication, she's put symbols on all the different localities that she visited, whether there were any musculister there, whether they were washed in small numbers or large numbers, or actually living intertidally in large numbers or small numbers, or well established offshore. This is the sort of work she was doing in the earlier part of her career. Now here's another part of documentation she made of the introduction of Mytilus galliprovincialis to northern New Zealand. Already we did have a few scattered specimens living here prior to the arrival of a northern hemisphere strain that her work showed arrived here in about 2006. And subsequent DNA work has shown that it came from in the northern hemisphere and maybe started hybridizing with the local blue mussel. But before its arrival, there were only a few scattered specimens found. You can see all the crosses where she surveyed the intertidal over many years, and they weren't there. And suddenly they started appearing in 2006 and spreading all around Waiheke and the start of Waitemata Harbour, as you can see here. Here's another marine invaders, two marine invaders, in fact, introduction to New Zealand of two sea squirts and their subsequent dispersal, published in 2009. The first was the club tunicate, which again, with this kind of map, crosses show when and where it wasn't found, and documented arriving here in the Auckland region about 2002, and then spreading again all around much of the outer Waitemata Harbour. Down here, we've got another tunicate, Punjavoy, which may have arrived in New Zealand at near North Cape up here about 1990, first at Twilight Beach in 2007. And subsequent surveys have shown it had spread around much of this North Island. In a different vein, Margaret contributed to understanding a marine invader to the Californian coast of North America. She collected specimens of this Philene from her local piece of seashore there at Farm Cove in 2005, and sent specimens over to the States where they did DNA sequencing and were able to show that many specimens that had been invading the west coast of the US were in fact the New Zealand species, Philene auriformis. So she helped them understand where their marine invader comes from. Between 1994 and 2015, less formal intertidal surveys were expanded to cover all of the Auckland coast. She and I went out every month on the spring low tide. And you can see here in red dots the different places we went to in 201 low tide surveys during that period, mapping the habitats, the zones, and also recording the biodiversity of these places during a spring low tide. And this sort of work led to two of her major summary publications near the end of her life on micromolluscs and nudibranchs, two of the areas she really enjoyed and specialized in, and that Wilma's certainly telling you about the micromolluscs. So first of all, the nudibranchs, and she really loved nudibranchs from an early time in her marine career, and everywhere she went, she was looking out for unusual and tiny marine nudibranchs or sea snakes. Here you can see one of them inside a limpet shell that she's holding a little bit of water that would come out for her. She recorded their habitats, their characters, their biology, and she made color illustrations of them for 40 years. because once she'd collected them, she did, then they would shrivel up and lose their colour and almost impossible to identify afterwards. So she made these colour illustrations of most of her collection. She collaborated with nudibranch specialist Michael Miller and Richard Willen, who helped her identify them in exchange for information and observations. And here you can see some of her black and white pencil illustrations of these nudibranchs that she saw and she was collecting or recording observations on. And here's some photographs, some of these lovely sea slugs around the Auckland coast. Here's one from Milford Beach. This one here was on the Huia coast, Parengarenga, and the sea hare, Timuri. And 4 more, Neola Reef, Waiwara. Look at this beautiful nudibranch from Red Beach, 2 of them here at Takapuna Beach. You can see why she fell in love with the sea slugs. Not that there are many people who fall in love with sea slugs. And so she prepared a summary publication, which was published here at the top. And here's one plate out of that publication showing her distribution records of 4 of the nudibranchs around northern New Zealand. So if you look here, the black diamond, for example, are her records intertidally of Giruna and Tharina, here, here, and up here. And so it's quite a rare species, whereas you can see a more common species is the red Rostangia muscula, and you can see she's got lots of records both on the west coast and the east coast, and extending right up to Parengarenga. Here's 4 more plates out of that publication, just 4 of her 24 plates, all like this, documenting her intertidal records, 78 species of sea slug from around northern New Zealand she published. Several of her finds were additions to the New Zealand fauna. In other words, they'd not been recorded from New Zealand before. They'd arrived here from somewhere more tropical up to the north or northwest. In this summary paper, she recorded 78 species, 76 of them occurred on the east coast. Only 2 species were confined to the west coast, with a total of 34 from the west coast. The most diverse area was around Lee, where she had 47 species. More than half of the records from northern New Zealand nudibranchs have been recorded from around Lee, 42 from the Bay of Islands, and 38 from Great Barrier. And in the harbours, she had 33 species from the Waitemata, 27 from Parengarenga, right here in the far north. And on the west coast, she had 24 species from the Manukau. No nudibranchs have yet been named for her, but they should be, and I recommend that to whoever's working on nudibranchs, if they have a new one from New Zealand, maybe Margaret deserves to have it named after her. She has, however, had 1 species of foraminifera named after her, Ammonia Morley, from a sample she collected in New Caledonia in 2001. This is, by the way, 0.1 of a millimetre here, so 0.3 of a millimetre across. Margaret Morley's micromollusc work, I'll summarise very briefly here. She published many papers and records and observations on the micromolluscs over a 40-year period, and then she summarised it in 2016. Margaret's field equipment, as I noticed, included a kitchen sieve or quite often a tea strainer, which might seem a bit odd, but here you see her using it. She would take it into her cap, pull it out when she found a place of fine-grained sediment. She'd dip into the watery sediment and sieve all the mud and sand out, and she'd be left with just the large micromolluscs, probably coarser than about 2 millimetres. And then she would put them in a bag and repeat the exercise a few times over to get the micromolluscs out. She also had a large plastic bag, and often out in the field with her at low tide, you'd see her sitting down on the job. You wonder, what are you doing, Margaret? And she'd say, I'm washing these rocks, their bottoms are dirty. And she'd lift up a boulder, and underneath the boulder would be a lot of micromolluscs tapped and grazing the underside. And she would wipe her hand over the bottom with the bag underneath. And all these little molluscs would drop into the bag for her to take home, put under the microscope and identify. She'd also always collect a big bag full of seaweed, put in a bit of seawater, and then she'd tie up the top and she would shake and shake and shake. and all those micromolluscs that were grazing on the seaweed would fall off and collect in the bottom of her plastic bag for her to study at home. So this is the one of the diagrams out of the publication in 2017 by Geography and Biodiversity of Micromolluscs in the intertidal of northern New Zealand. In that particular publication, she records 367 species, all smaller than 10 millimeters from the northern New Zealand intertidal. And at least 154 of them were living intertidally. Many of the others may have been washed ashore in storms, etc. 361 she recorded from the East Coast. That's only 6 species in northern New Zealand that are restricted to the West Coast in that study. And she had a diversity of 157 species on the West Coast. Here we see a plot that shows the diversity at different sites, the number of species at all these different sites around the Auckland region. And you can see that the greatest diversity is out in the outer part of the Hauraki Gulf, where she recorded between 60 and 140 species. Some of these localities you can see here from Waiheke, Tripoli, on Great Barrier, and up here north of Putakaka. Whereas the diversity of micromolluscs drops off as you get into greater shelter, greater suspended sediment into the Waiheke Harbour, or the upper Root Pacific, and the Manukau, as you can see here, with fewer than 10 species. And even on the wild West Coast, very low diversity too, she recorded. Here's one of the plates out of that publication. She recognized a number of different associations after doing cluster analysis, etc. And this is the localities where some of those common species in the shallow water occur in the reds here. So the Nucula hartwiggiana, or Arthritica bifurca, or some of these species here. And here she is crawling along the high tide line looking for micromolluscs with a little dog written behind. These are just some more of her plates from that publication, showing the distributions around northern New Zealand of some of these micromolluscs she was recording, and all her beautiful illustrations, as you can see. So Margaret Morley, besides these 2 summary publications on her specialty areas of micromolluscs and nudibranchs, also took part in many group surveys of certain areas. And she was an integral member of those groups, doing the surveys, and she did the identification of the molluscs, and later on in the seaweeds. So she did worked with teams documenting the marine biodiversity of Ringaringa, Ahapana, Hayward Island, Taranaki, Kawarua, Great Barrier, Whangapua, Imakuka Bay, Waitemata Harbour, subtidal, middle Waitemata Harbour, intertidal, Waikato, the Manukau, Tamaki Estuary, Raglan, Kawhia, North Taranaki, New Plymouth, Mahia, Auckland Islands, and a number of other places as well, all of which were published with her as a major contributor. Here she is in 1993 on one of those visits to Kawarua on the west coast of Northland. And here's Jenny Riley, who did actually catalogued all her collection that Wilma was telling you about on that trip in 1993 with Glenn. Here's Margaret in the field. Here she is photographing one of those giant introduced Japanese swimming crabs, Charybdis japonicus, here wanting to have a nibble at her. And here's another photograph of Margaret's work. Mostly she identified the molluscs as her major role, which was the Stupus, but in 2000 she added the identification of seaweeds to her repertoire. And from then on, she was the major contributor to the identification of seaweeds from all these surveys around the coast of northern New Zealand. And here's some of Margaret's seaweed illustrations that we used to illustrate some of those publications. One of the major surveys that she was an integral part of, as you can see listed third in the co-authors there, was faunal changes in the Waitemata Harbour sediments 1930s to 1990s. It was a study undertaken at the Auckland Museum, where Powell had published on the biota of the subtidal sediments of the Waitemata harbours in the 1930s, and in the 1990s, a group of us decided to go and resample Powell stations and add a whole lot more. So the open triangles and circles are the samples of of Powell that were resampled, and the black squares are the new stations that were also sampled during that time. And the samples were brought home, and Margaret went through many of them and identified and counted the live molluscs, which contributed to the results of the study. And it was published here in 1997, and this is Powell's map of the distribution of marine associations back in the 1930s, and that map produced in the 1990s. The conclusion was that 14 mollusk species had disappeared or greatly reduced in their occurrence in the Waitemata Harbour since the 1930s, and that 3 introduced bivalve species had now arrived and become dominant in the harbour sediment by the 1990s. These were the date mussel, Pelista, the small bivalve Fiora lubrica, and another bivalve Limeria.
We've lost you, Bruce.
Yeah, somebody muted me. Try again. Okay.
I'll just say, Bruce, sorry to interrupt, but your sound is quite muffled at times and hard to hear.
You're muted at the moment, Bruce. Yep.
I'm right now?
Yep.
That did it itself somehow.
Too, Bruce. I don't know quite whether you've got blurred background, but there's a bit of blurred foreground too.
Hmm.
You've got sticky tape on.
No.
Oh, there we are. That's better.
Hmm. I don't think that makes much difference except to my background.
No, that's good. That's better.
In in the late 1990s, Margaret and I and others moved on to mapping and undertaking surveys of the intertidal life around the middle and upper Waitemata harbours. Added that to the subtidal work we'd done. We mapped the habitats and documented the biodiversity and produced it as a report by the Auckland Regional Council. There you see the map of the habitats and some of the more common biota around the Waitemata Harbour. In that publication, 724 species were recorded living in this marine area, and Margaret made a major contribution to that documentation. What's going on now? Now my screen has decided to freeze up.
You're going fine for us.
Yeah, oh, there we go. Okay, then we moved on to the Waitakere Ranges, and in 2004 published a report on the intertidal life around the west coast of the Waitakere Ranges, and that also was published by the Auckland Regional Council. During spring tides, we progressively mapped and walked the entire coast of the Waitakeres, documenting the biota that was present, the intertidal, and mapping the habitat and the communities there. Nearly 500 species were recorded living around the Waitakeres coast in that particular study. And then we moved on to her local strip. This is where she lived for much of her life here in Pakuranga, where my pointer is, and we mapped the habitats and communities and documented the biota of the Tamaki estuary and intertidally there. And it too was published as a regional council report. And in the Tamaki and just the entrance there, we recorded 390 species living in this area. Here's some of the legend here, mapping different kinds of rocky substrate, sediment substrate, vegetation, and faunal elements in that particular map. I thought I'd tell you about one of Margaret's most intense research programs or projects, one she took on in 1997 through to 1999. It was undertaken in the Hobson Bay mud. You all know the Hobson Bay, and I'm sure you've all had fun in the mud there. Not many people ever venture forth, but we did. And every month on the spring low tide, we'd go out into the mud and we'd take replicate samples of 0.5 of a square metre area of mud, digging it down to 10 centimetres and putting the mud through a sieve like this with a 1 millimetre opening. This captured all the small specimens of live Fiora so that we could monitor their growth and study. this introduced small bivalves life cycle. So this was a study over a 2-year period to study the life cycle of the introduced Japanese bivalve. And these are the results of that work. Margaret took all those Theora home and she measured every single one of them, the length of them, and counted up the number in 1 millimeter size lot. So on each histogram here, we've got 1 to 2 millimeters, 2 to 3, 3 to 4, 4 to 5, and 6, and the number of each for each month. And as you can see, in some months you had to count 860 small bivalves and measure them. And that produced these histograms through 2 years, starting in September 1997, passing on up every month through to August 1999. The result showed considerable variation in the density of this introduced small bivalve that was so common around the Waikato harbour, between 30 and 850 per square metre. So over here you can see there are only 32 recorded in here and 800 odd in March a year later. We recognise 6 cohorts, that is populations that became established and we could monitor growing through time before they died off. A, B, C, D, and F. So here we can see it small, and you can see through time they're gradually growing bigger. Particularly you can see it in these ones through here, another cohort here, and another cohort going on through here. So there were 6 cohorts we recognised, and in cooler months, that's these ones up in the top here, sort of April through to August, there was slower growth rates. As you can see here, they're growing much more slowly through the sub-cold months, 0.5 of a millimetre per month. And they lived a lot longer, 6 to 8 months. And in the warmer summer months, as you can see through here, they were faster growing. They could grow at 2 to 3 millimetres a month and had shorter lives, as you can see here. So all that measuring by Margaret of these little bivalves we got out of the mud produced a very nice study of the life cycle of this little introduced bivalve. But we wondered, why was it that in one summer here in 1997, 98, were very low numbers of Theora, and yet in the next year there were very high numbers, as you can see here. We noticed there was a correlation between high mud crab numbers, 40, 40, 40, 40, and low numbers of Theora, or between low crab numbers, 8, 1, 6, 1, and very high numbers of Theora. And so we think it was the crabs that likely were eating off the Theora as they were becoming established and growing. We did a controlled feeding experiment, showed that one mud crab, Almus hirtipes, consumed up to 7 Theora per day in our little captive tanks. So we felt confident that that's probably an explanation for the low numbers. And we did another sampling the following year in summer there, where we again had very low numbers of Theora and again very high numbers of mud crab, 74 and 38. In 2005, Margaret was looking for another challenge, and I suggested she look at the poorly studied modern marine ostracods. I said to her, just like microscopic bivalve molluscs, and could complement our foraminiferous studies. And the first of her studies on molluscs was published in 2006. Margaret's ostracod research in just 10 years resulted in 8 papers on ostracods, which added 92 new ostracod species records to New Zealand. That's ostracod species that have never been recorded from New Zealand, Margaret found and identified. 45 of these came from the Hauraki Gulf, 30 from the valleys, and 17 from Mackay Bay. And here's just some of Margaret's magnificent pencil sketches or drawings of the ostracod she found, almost all of which were published in various publications, and so document the ostracod biota of northern New Zealand, which no one had done previously. Here's a figure from the next paper, 2007, on the intertidal and shallow water ostracods of the Waikato Harbour. You can see the stations that she looked at here between intertidal and 30 metres depth. And in that small area there, she identified 88 species between Hobsonville and Rangiputo, and 5 associations, from associations in the low salinity head of the harbour, species like this, up to the subtidal with quite different associations. A bigger study was published in 2012, where she studied 82 samples from the Hauraki Gulf, a depth range of 0 to 130 meters. And in that study, the diversity bloomed out 154 species, and by numerical taxonomic work, by analysis, computer analysis, she recognized 10 associations that we see up there above. And these are the different species that characterize the different associations. You can see some are tidal associations right through to a deep water association with a mean water depth there of about 60 meters. Here's some of her beautiful line drawings again. These are some of the additional species for New Zealand that she got from the Cavalli Islands. 30 in all she got there. And these are some of the species that you'll see many of them she wasn't able to recognize to any described species name. So they're sitting there waiting for another ostracod expert to come along and eventually describe a name. Although you can see one or two she's going to link to describe and name Australian species. She did another detailed study on the tidal zonations of ostracods and micromolluscs on the east and west coast of Auckland that she published in 2010. This also required a great deal of work at home. These are her two sample sites at Music Point and out on Puponga Peninsula for molluscs. She took replicate samples of 25 cubic centimeters of corallina earth, seaweed from high, mid, and low tide. 6 samples from each of these sites at 12 samples. She then did a complete census of all the ostracods and all the micromolluscs at each and recorded the live and dead specimens separately. She had 80 species of ostracod and 81 of micromolluscs in the study. And these are histograms of her ostracod results. As you can see from here, they've got the west coast and the east coast plotted and high, mid, and low tide over here. These are the histograms of the number of these different species in each of the different sites. And she concluded that tidal elevation was a strong driver of the distribution of low tidal faunas, which were more different from the high and mid tidal faunas in the ocean. You can see that quite a big difference here and here, the species here and not in the mid and high tide. She found there was a large difference between live and dead, between these lots and over here, the dead fauna, especially in the low tide here and here, reflecting the post-mortem transport in of ostracod carapaces by waves of time. So there was a lot of during storms, dead specimens from offshore were thrown up at low tide and collected in the association of those that were living there. This is the micromollusc data from that study, and in that she concluded that the biogeographical differences in the east and the west coast faunas are much stronger in micromolluscs than in the ostracod. So we can see species like these which are occurring on the east, living on the east coast intertidally, and not on the west coast. So others down here on the east coast and not on the west. And similarly, large differences in the dead fauna are more diverse than before of the micromolluscs on the east coast than the west coast. And so that brings me to an end. This has been just a brief summary of the research of Margaret Morley. She did far more than I can cover in this time, but I'd like to summarize some of what I thought were her major works, especially in the last 2 to 3 decades, when she really put her mind and focus on doing and publishing the research. So thanks for listening. We can open it up to questions for Wilma, or for me, I guess. Wakey, Wilma.
Okay, just a reminder, if you want to ask a question, you'll have to unmute yourself, which you can do. I'll start with a quick question to Bruce. Off the top of your head, Bruce, how many GeoClub trips did Margaret do?
Over 200. I was reading, I haven't actually counted them. I don't have records quite as detailed as he went on almost every geology field trip between 1994 and 2015. Any more questions?
You forgot to mention her stick as part of the kit. She took her stick everywhere. I can just visualize her always with her stick on a beach.
Yes.
I'm not quite sure what she actually used it for.
She saved herself bending down sometimes. She used to flip shells over or flick things around with the stick.
And she would certainly defend herself against fierce crabs and put it above her head to stop the seagulls attacking.
Oh, yes.
I think she might have used it to measure the depth of the mud we were about to go through as well.
Or she could have possibly, but she certainly used it to flick things over and save bending down all the time.
Anybody else?
For Wilma also, if you have a question, now's your chance. You must have been too comprehensive, Bruce.
Oh, they've all gone to sleep.
Overwhelmed.
Yep.
I don't have a question, but I do have a sort of comment as to, you know, you said that, you know, she did all this work as a volunteer, so she technically wasn't a professional, but the impact that she had